So last class we talked about DNA replication. Today the focus is going to be on transcription, and we'll be looking at that in the context of diabetes and anabolic steroids. Diabetes mellitus, the term mellitus is Latin for sweet. It's a group of metabolic diseases that, as I'm sure many of you are aware of, that is characterized by hyperglycemia or high blood sugar concentration that can be a result of a few different reasons, either defective insulin production, defective or resistance to insulin, or it could be both. And so the appropriate question is, what does insulin do in the body? So what's important to know is that there are insulin receptors on our cells, and the binding of insulin to those receptors, what it does is it activates a series of transporters that helps to shuttle glucose in and mediate the conversion of glucose through metabolic processes that we're going to talk about in the next unit into ATP. And so the role of insulin is incredibly important in the ability to activate these receptors and to mediate the shuttling in of glucose as a key energy source, or to convert it to a cellular energy source. The chronic high blood sugar or hyperglycemia that's present within diabetes, as you can imagine, it's associated with a lot of clinical consequences, because if there's an inability to appropriately shuttle in glucose, those cells will experience energy deprivation and subsequent cell death. And so some of the consequences of this hyperglycemia can be the failure or dysfunction of of various organs, especially the eye. That's sometimes described as retinopathy. There are these thin blood vessels across that are present within our eyes, our fingertips, and around nerves that, when lacking the appropriate nutrients, can die. And so this is true with our nerves, where there is neuropathy among diabetes patients, and as well as there's this reality that with high sugar content in our capillaries, the blood flow, the viscosity is going to change, and subsequent blood flow is going to be altered as well. The symptoms of diabetes, polyuria means that there's frequent urination. What's going to end up happening is that with this high sugar concentration within the blood, at the nephron, at the site where there's ion exchange, there's going to be, with an increased sugar concentration, there's going to be an osmotic gradient or osmotic pressure that's going to drive lots of water to follow sugar into our urine. And what's really unfortunate for those of you who aspire to be clinicians is that a really old method for determining whether someone was diabetic was to see how sweet their urine was. And so there's the sugar goes across into the urine, and there's an osmotic gradient that shuttles water in across, and so that explains why there's so much urination. And as a result of that water loss, there's also polydipsia, a constant thirst. And so in addition to that, as I mentioned before, that there's going to be a lot of nutrient deprivation in a lot of our cells because of the inability to shuttle glucose into these cells. A lot of patients, before they realize they're diabetic, will experience a lot of weight loss. In addition to that, again, there's nutrient deprivation. It's going to also lead to impaired growth. And there's also going to be what we call ketoacidosis. And so in the absence of glucose as an energy source, our cells will compensate and look for other molecules as energy sources. And so one way in which that can happen is another pathway where it's going to look to either proteins or lipids as an energy source. With lipids, there's a process called gluconeogenesis. This is not important to remember, at least right now. It's the breakdown of protein to try and form glucose to try and create that energy source, and you're subsequently going to be losing muscle mass, losing protein. For ketoacidosis, that's going to be using lipids as the energy source, whereas they're metabolized, they're going to form these ketone bodies or these ketones. And as these ketone bodies are being formed, they're going to be released into the urine. And so in addition to that, there's this fragrance when people are in a state of ketoacidosis. Their urine will also become a lot more fragrant as a response to these ketone bodies being excreted. The long-term complications of diabetes, as I described before, there's retinopathy, the fine capillaries within our retina, those capillaries, the cells within the capillaries, and those endothelial cells will begin to die off, and you'll have subsequent blindness. And so that's one late-stage consequence of untreated diabetes. There's diabetic neuropathies. Their nerves will also begin to experience that nutrient deprivation, and as a result, their nerves will begin to fail them as well. And in addition to that, this is a metabolic disease. There's also subsequent cardiovascular and cerebrovascular diseases as well that's associated with type 2 with diabetes. In addition to that, there's altered shuttling of lipoproteins or lipid-associated proteins. And in addition to that, there's also a periodontal disease that's associated with this as well. Typically, what ends up happening is that there's, in the presence of this excess of sugar that's not being used by our own cells, bacteria love sugar. And so the thought is that these bacteria end up taking residence within the oral cavity and becomes a site for bacterial growth and bacterial infection. The prevalence and economic impact. So this is going to be one of the diseases that has probably one of the highest economic impacts that we'll come across. So I tried to find the most recently available information. This is as of 2017. There is $327 billion in total cost for diabetes, and for direct costs associated with actual treatment and care, there's $237 billion alone. It's typically been, there's a trend of about 33% increase over every 5 years in the total cost. And so that means you can increase each of these by about a third. And so as we have an aging population as well, oftentimes as people age, as cells become senescent, as we talked before, everyone in their old age, for lack of a better phrase, becomes diabetic in one way or another. And so what's really unfortunate is that the incidence rate right now is 2 million per year become diabetic. That number is probably going to increase. But what's been really interesting is that historically, once you become diabetic, it's irreversible. But what's been really interesting is that there's been a lot of therapies that have demonstrated that with really careful diet and exercise, and I can attest to this with personal anecdotes of people I know, that they can actually, their severity of diabetes or the requirement for drugs can actually go away with appropriate exercise, with really careful and regimented exercise and diet. It can actually begin to go away, but that's anecdotal, so take that for what it is. So the prevalence rate, this is typically rare, has usually been rare in African and Asian nations, and that can be usually attributed to the diet that's present within a lot of these nations. But what's been unfortunate is that as what we dub the Western diet, as the Western diet has been emerged in a lot of Asian and African nations, as evidenced with more KFCs and McDonald's in a lot of these countries, the rate of increase of diabetes in these nations are actually highest because of the Western diets that have been adopted there. This can also vary by race in the US, but it's typically on average about 8% by each race. And what's interesting is that there's a genetic predisposition that exists among some populations, where some populations have up to a 50% incidence rate, including the Pima and Papago Native American tribes. Are there any questions so far? All right. So for the cause, as many of you probably know, there are different types of diabetes. Type 1, which is historically dubbed juvenile diabetes, Results from the loss of the insulin producing cells. Those are the beta cells within the pancreas, and that it's actually due to an autoimmune disease or autoimmune destruction of the beta cells in the pancreas. They present with an autoantigen that is typically an autoantigen that's present on the beta cells that leads to their destruction and leads to the total absence of insulin. And so that's why typically patients will typically present with type 1 diabetes between 6 to 12 or 6 to 13 years old, and they're usually treated with just receiving insulin shots. Type 2 diabetes, which was typically referred to as mature onset diabetes, has a different pathogenesis and is a much more prevalent category. It typically results from the it kind of branches off into two different subsets, but it can either result from either the improper regulation of insulin or the improper insulin signaling or resistance to insulin. And so one example is mature onset diabetes of the young, which is not an autoimmune disease, but rather a transcriptional defect that's inherited. And so the MODY, or mature onset diabetes of the young, is one subset of type 2 diabetes. The other is insulin resistance, which is typically what we associate with poor diet and poor exercise. That also is what typically also developed in old age as well. And so it's a result of them being desensitized to insulin because the blood sugar is so high, where usually our blood sugar level is right at 100. Oftentimes it can be as high as 400 in those who become insulin resistant. And so we're going to use mature onset diabetes of the young because of its transcriptional defect as a way to try and use it as our backdrop to understand transcription, or at least apply transcription in diabetes. And we'll also be using that with anabolic steroids as well. But before we jump into transcription, are there any questions about diabetes at all? Yes. Right. Yeah. MODY is a transcriptional defect. Type 1 is an autoimmune response to autoreactive T cells. And before I forget, one last one that's not mentioned here, it's historically dubbed type 3 is gestational diabetes. That's typically when women who are pregnant, who did not have diabetes before, can become diabetic during pregnancy. It's not entirely well understood, but a working understanding of it now is that there's so many hormonal changes that occur during pregnancy that there is a hormone imbalance. Again, insulin is an endocrine factor, that these other hormonal endocrine factors can disrupt insulin signaling that creates issues that creates a hyperglycemia within these pregnant women. And what's interesting, this is the case with my wife, there can be pregnancies where it doesn't occur, and then pregnancies where it does occur. And usually after that, it occurs with each subsequent pregnancy. Are there any questions before we jump into transcription? All right. So before we talked about DNA replication and how DNA polymerase, the different versions of DNA polymerase participate there. And today we'll be the polymerase we'll be focusing on is RNA polymerase. So there's the key area, or a term that we use when talking about DNA or genes or the genome is the term for site is locus. So the locus that you're focusing on, where there's a gene that's going to be transcribed, that's called the transcription unit. So in eukaryotes, transcription unit equates to one gene. In prokaryotes, typically it can correspond to multiple genes that are in tandem with one another. But in eukaryotes, one transcription unit equates to one gene. And the site where transcription begins is at the promoter, and the site where transcription ends is called the terminator. So what ends up happening is that RNA polymerase will be recruited to and bind to the promoter region of the gene. And unlike DNA polymerase, RNA polymerase does not require the accessory proteins that DNA polymerase does. And also what's important to know is that, if you think of if we were to think of the phone cord as our surrogate for of the DNA double helix, we talked about how they're supercoiling when DNA polymerase and the helicases unwind the DNA. Here, what's happening is that RNA polymerase is sliding in, and there's no supercoiling here, so there's no accessory proteins that need to address that supercoiling. So RNA polymerase binds to the promoter region, and and what's important to also note is that the elongation that occurs is from 5' to 3', just like with the DNA polymerase. So all of our polymerization events, or the synthesis of RNA or DNA, is occurring in a 5' to 3' direction. So what that means is that the RNA polymerase is recognizing a template strand at the 3' end. And so the template strand is what the RNA polymerase is using to encode its RNA transcript. And what's really convenient is that typically with the template strand is anti-parallel to the RNA that's being synthesized. And that RNA that's being synthesized will match pretty nicely with what's labeled here as the coding strand. The only difference will be that instead of T's, there will be now, instead of thymines or T's, there will be U's or uracils on the RNA strand. And so the RNA transcript and the coding strand will match up, and the template strand is what's being used to encode or transcribe the subsequent mRNA. And so this elongation happens pretty quickly, and RNA polymerase, as it goes along, is using ribonucleotide triphosphates, or NTPs, as the monomers to help create the mRNA transcript during transcription. And so as it elongates and continues to create this mRNA transcript, RNA polymerase is going to reach this terminator that has this termination code encoded in that site of the DNA that'll then prompt both that code and what happens to the RNA transcript for RNA polymerase to release and undock and leave the DNA and end transcription. And you'll be left with what we call the pre-mRNA. Are there any questions there so far? So this is just kind of the big picture, and what we're going to do is we're going to dive a little bit further to fill in the details here. And so we have RNA polymerase that, different from DNA polymerase, does not require accessory proteins to unwind or address supercoiling, because there's no supercoiling here. We have a promoter region that is the start site, a terminator region that's the stop site. So what we're going to do here, a little bit different from replication, we're going to, instead of going back and forth and talking about eukaryotes and prokaryotes, we're going to talk about prokaryotes or bacterial cells a little bit, and then see what this looks like in eukaryotes. So similar to, as we mentioned before, I mentioned in the previous slides, we have a template strand and a coding strand. Our template strand is what RNA polymerase is reading and using to encode and create an anti-parallel mRNA transcript that, again, will match the coding strand, with the exception of, instead of T's, we'll have U's. And so what's going to end up happening is that the promoter is where RNA polymerase 2 will bind. And these promoter sequences will also has what we talked about previously, these consensus sequences that help encode that this is a site where transcription will begin. And so when looking at different versions or different organisms and looking at their looking at their promoter regions, one of the key sequences to note is that there's a TATA region within the promoter. We call it the TATA box. And we'll be talking about a little bit about how that contributes to initiation of transcription in a moment. And so when RNA polymerase binds to the DNA here, what's going to end up happening is that it's going to stay for a little bit. And what's happening there is that as it binds to and docks on the DNA, what's going to end up happening is that at that site where transcription is being initiated, the first 9 to 10 base pairs are being added or being transcribed. And then after that occurs, and it's using these nucleoside, the ribonucleoside triphosphates as the monomers to create the mRNA transcript. And after those first 9 or 10 are being created, then it'll begin elongating, going down across the DNA and producing the mRNA transcript. And doing so in a 5' to 3' direction. And so this process of moving down in a 5' to 3' direction that it's synthesizing, we call that the elongation step. And so the elongation is what it's doing is it's unwinding the DNA, able to go in and read the template strand and create the subsequent mRNA transcript. And it's using the monomer ribonucleoside triphosphates to create the mRNA transcript. And so what's going to end up, and just to remind you guys again, there are no accessory proteins during this elongation process. There's no helicases or gyrases being used here. Yes. During initiation, when RNA polymerase II docks onto the DNA, there's going to be the first 9 or 10 base pairs that are being added, and that RNA polymerase II, when binding to the DNA, does not move at first; it stays put, but then continues to go along. And so what ends up happening now is at each site where the RNA polymerase II is present, like if you were a snapshot in time, there's about 12 base pairs being added at each site that it's at, if that makes sense. Yeah. Yes, based on its, by sheer nature of the size of RNA polymerase II, it's able to add that many base pairs at a time when creating the mRNA transcript. So the step that ends this transcription, we call termination. And so the terminator sequence is typically what encodes the signal for transcription to stop. But what's really important is that when we talk about, this is true for replication as well, when we talk about these processes, this is happening incredibly quickly. It's hard to envision that because whenever we're talking about this and looking at a slide or a moment in time. But if you were to try and I can probably try and send out a video of this, but if you were to try and see this in real time, this is happening incredibly quickly. And so what's important is that there needs to be signals that can help stop transcription so that when genes are being transcribed to try and encode corresponding proteins, that you're not having different genes that aren't meant to be together separated, so that you don't have weird protein dimers or complexes that aren't supposed to happen in nature. And so what ends up happening is that we have these within the mRNA transcript that's being produced during transcription, there's going to be this self-complementary nature of it. And so as it's being synthesized, there's going to be regions encoded within the terminator sequence that are rich in these CG rich regions. And so what's going to end up happening is you're going to have, for example, you're going to have a region that might read CCCCCCC and a subsequent region that reads GGGGGGG. And those self-complementary regions are going to fold over and bind and create this hairpin loop. This hairpin loop is going to be incredibly important in providing a biophysical signal to the ribosomes and RNA polymerase II to dissociate from the DNA to stop transcription. And so this hairpin loop is a bit of a secondary structure that exists within RNA. And so this is kind of similar to the structure, the primary and secondary structure that we talked about with amino acids and proteins, where the primary sequence would be just the specific nucleotides within the mRNA transcript. And this secondary, the hairpin loop is a secondary structure that occurs as a result of the self-complementary regions that are a result of the terminator region. And so this hairpin signal, because of either ways in which it biophysically interacts with local ribosomes or intermediary proteins that can communicate with RNA polymerase II to stop and halt termination. Are there any questions there? Yes. Yes. Yes, exactly. Yeah. Yeah, this structure is it's a biophysical cue for the termination of transcription. And this is true in both prokaryotes and eukaryotes. Yes. Is there a way to the rest of the RNA between the hairpin and like the... We're gonna get there. Hold that thought. That's a great question. All right. So to focus more specifically on eukaryotic transcription, we have 3 RNA polymerases. And we have one, and each of those polymerases correspond to the type, a type of RNA. And so for RNA polymerase I, that helps with the production of ribosomal RNA. RNA polymerase II, as designated in blue, is the most important one that I want you all to take note of, is what helps make mRNA, messenger RNA, and that's the RNA that encodes for proteins that will be used during used and encoded during translation. And finally, RNA polymerase III is what's important for transfer RNA. And so each polymerase also recognizes a different promoter structure. And so there's going to be features of the promoter that help encode for what specific RNA polymerase is going to be recruited. So for the initiation of transcription, that occurs at the promoter region. And so RNA polymerase II is RNA recruited to that promoter region. Is that we have this initiator sequence that's around the start point. Let me see if I can use my mouse here. So this black rectangle is going to be the transcriptional start site. And the numbers that you see, this +1 or -25, correspond which direction from this transcriptional start site we're moving. And so it is this transcriptional start site is a single nucleotide at which things will begin. And so upstream from that, in the we're looking at the coding strand here. Upstream from that, in the 5' direction, there's going to be what we described before, the Tata box, this consensus sequence that is responsible, that encodes for the initiation of transcription. And so both the Tata box and the initiator of which this transcriptional start site is a part of, and the start point is actually bounded by specific nucleotides that can either be 2 pyrimidines, which are thymidine or cytosine, and two to the upstream direction and 5 downstream in the 3' direction. Both the Tata box and this initiator sequence make up what we call the core promoter. We have what we call the B recognition elements upstream, and we have these downstream promoter elements. Those are less important, and as you can tell, are much longer and have a little bit more variability there. But the main part that I want you to take note of is the core promoter that makes up the Tata box and the initiator sequence. And what ends up happening is that there's this class of proteins called transcription factors that will actually bind to the Tata box, physically bind, and recruit RNA polymerase II. And so these transcription factors come in different flavors, but the main ones that are important for those that bind to the Tata box are called basal transcription factors. That are always required to allow RNA polymerase II to bind. And so for RNA polymerase II, the main one to take note of is we call it transcription factor 2D or TF2D. And it is, again, the basal transcription factors are a general class of transcription factors that bind to the core promoter. And what ends up happening is that there are other transcription factors that can come in and bind to TF2D. And now these other transcription factors are regulated differently. So for TF2D, if the Tata box is available, it will bind. It will bind to the core promoter as long as it's available. And we've talked about before how segments of our DNA are regulated based on their physical openness and availability. And so if that core promoter region is available, TF2D will bind, and there will be other transcription factors that will come in and create this multi-molecular complex, almost like a scaffolding site for RNA polymerase II. That's called the pre-initiation complex. These other transcription factors that come in and bind with TF2D, they're all regulated differently. They can be regulated either by phosphorylation, they can be regulated by spatially where they are by being shuttled from the cytoplasm into the nucleus. But what's important to know is that it's not only TF2D, this basal transcription factor, this like general class that just binds to the core promoter if it's available, but rather it requires these other transcription factors to come in and bind and create this pre-initiation complex that provides a docking site for RNA polymerase II to bind to the promoter. And so what that looks like is TF2D here will bind to the Tata box as part of the core promoter. You'll have these other transcription factors that will bind in and create this pre-initiation complex. And once that's formed, you'll have RNA polymerase II. And again, we have our black rectangular box, which is the transcriptional start site. And once RNA polymerase II is there, you'll begin transcription and elongation as it moves down. Before I move on there, I want to stop and I want a couple questions. Yes. Yeah. Yes, that's correct. And so that makes up the initiator sequence. Not which promoter. So this is the promoter, not which promoter is going to bind to it. So this will recruit with the presence of transcription factors, recruit RNA polymerase II. Are there any other questions there? One more question. And as I said before, I love dumb questions, so I'll take a dumb question too. I don't want to call them dumb questions. I like to call them more foundational questions, but I want to give space for you all to ask anything. All right. So, yes.
So does the core promoter become a part of the new RNA, like the new pre-mRNA?
That's a great question. It does not. It's not part of the mRNA that's going to be synthesized. It is a site that signals. Actually, if I go back one slide, that'll actually help. So the black box here is where transcription will actually occur. That's the first nucleotide. So the Tata box is upstream in the 5'. Again, this is the coding strand. So 5' to the left, 3' to the right. In the upstream, the 5' direction, that's where the Tata box is. And the initiator sequence flanks the transcriptional start site. So only a few of the nucleotides will be present on the mRNA transcript, but most of it will not be. Most of it is upstream from the transcription start site and will not be part of the mRNA that's synthesized during transcription. That's a great question. So, for example, here, the last nucleotide part of the Tata box is in the -25 position. That means it's 25 nucleotides upstream from the transcription start site. Everything downstream to the right of the black rectangular box, the transcriptional start site, everything to the right downstream will be part of the resulting mRNA transcript that's synthesized. That was a great question. Okay, so now we have our pre-mRNA, or for lack of another phrase that's oftentimes used, our nascent mRNA. And so this primary transcript is going to be modified in a few different ways. And each of these modifications are going to either contribute to the resulting protein that is produced during translation, or modifications that help with the stability. I'm sure many of you probably heard with the mRNA vaccines for COVID that there's a question of stability or a question of handling. That's typically because mRNA or RNA is easily degraded. And so what's important is that there are modifications that help with stability of mRNA. The first that we're going to talk about is called a 5' cap, or 5' methylated cap. What this is that it is a backwards cap of a methylated guanine. And the reason why it's backwards is because we talked about how everything is 5' to 3'. This 5' cap is actually going to be a 5' to 5', the 5' carbon having a phosphodiester bond to the next nucleotide on its 5' end. And so typically, if you keep looking down, we have the 3' end binding to the 5' end, 5' end. And it's like everything's moving in a direction. There's a directionality, but this cap is going to have a 5' to 5' phosphodiester bond. In addition to that, there's a 7-methyl. This is a 7-methyl guanosine. What that means is that on the seventh carbon, on the 7th position, there's a methyl group on guanosine here, and it's added during elongation. And what this is, it's a really important signal that works in a few different ways. What ends up happening is that it's actually going to promote the recruitment of these cap binding proteins. And these cap binding proteins are going to help facilitate the nuclear export of the mRNA out of the nucleus. And in addition to that, these cap binding proteins are going to physically prohibit or stop the degradation of this mRNA. Again, mRNA is going to be easily degraded. And so these are evolutionary conserved proteins that help promote the protection of this mRNA so that it might be shuttled out for translation to create the important proteins that it's encoding for. And in addition to that, this 5' cap helps to promote a binding signal for ribosomes during translation. So this 5' cap is catalyzed by what we call a capping enzyme. There are a few different capping enzymes, and we're not going to get bogged down with the details of these different enzymes, but rather that what's important to note is that there is an enzyme that mediates this 5' backwards cap that helps mediate all of these functions that I just described. And it only associates, this 5' cap only associates with RNA polymerase II. And that's important because this is important because you only want this to happen with mRNA as opposed to ribosomal RNA or transfer RNA. So that's on the 5' end. We have this backwards 5' cap on the 5' end. Now looking towards the 3' end. Generally, these transcripts are kilobase pairs long. They're incredibly long, and they'll vary based on the size of the transcription unit. And so what ends up happening is that there's an enzyme called a poly-A polymerase that finds this poly-A signal. So typically at the end of the, that's part of the terminator sequence, there's going to be a sequence that's encoded AAU, AAA, and that is a signal that provides two functions. It's a signal during the handling of this mRNA that this is the end of the important nucleotides that encode what's really important. In addition to that, it's also a site that is targeted by poly-A polymerase, or PAP. So what ends up happening is that at this site where we have these this conserved sequence AAU, AAA, and it'll cleave the transcript there, and then it'll add a poly-A tail, a lot of adenine nucleotides to the end of that transcript. And this poly-A tail helps for a few different reasons. The presence of a poly-A tail recruits poly-A binding proteins that, again, helps with the stability of the mRNA transcript to stop it from being degraded. And it's also necessary for the full initiation of translation. And so the poly-A tail is added on the 3' end, is recognized, PAP recognizes the conserved sequence, and it mediates the addition of that poly-A tail. And what's important to know is that we have 4 different nucleotides. We have different nucleotides available at the RNA level. And the question, there's a natural question of why adenine, why A's. The idea is that with ATP, we have so many adenines available that it's more widely available than any other nucleotide, so that it's this conserved process of using those free adenines to in conjunction with poly-A binding proteins to help mediate the protection of the mRNA transcript. And typically the length of the poly-A tail differs based on what kind of cells they are. Prokaryotes can typically be about 30 nucleotides long. For eukaryotes, for us, it's typically 30 to 100. And so what's important to note is that we have our mRNA transcript with our poly-A tail that has our poly-A binding proteins. What's going to end up happening is that over time, there's going to be de-adenylation that occurs where the poly-A tail is slowly whittled away. And typically what's going to happen is that's almost like a timer, where basically if this mRNA transcript is not used, it's eventually going to be degraded. And so what that looks like is that over time, the poly-A tail will be whittled away, the poly-A binding proteins will dissociate. And what's going to end up happening is that in addition to that, this decapping protein will remove the 5' cap off. And now exoribonucleases can come in and totally degrade the mRNA. And so that's this de-adenylation and this degradation process is kind of a timing mechanism, and an endogenous timing mechanism to ensure that mRNA, we're not just being, our cells are not constantly bombarded with mRNAs that are residing around, but rather an endogenous mechanism to get rid of mRNAs. In addition to that, exoribonucleases are also used to help protect against RNA-based viruses. Before I move on to the next subject, are there any questions there? Yes. Mm-hmm. That's a good question. So this conserved sequence that is encoded by the AAU, AAA, what ends up happening is that this is a signal that everything downstream from here, and this is coming from the terminator, everything downstream from here is not part of the transcription unit. These are not nucleotides that are encoding the protein, but rather now we are, to anthropomorphize them, like we are nucleotides that are tasked with helping to end transcription. And now everything happening from here with the work of PAP, poly-A polymerase, is going to be cleaved away, and now a poly-A tail is going to be added on. And so like it's a sequence that denotes there's no special information here. Everything here is helped used for the termination of transcription, and now we can remove it and have a poly-A tail added on. Yes. Yeah. Ribosomal binding sites fit in with the hairpin loop. So the hairpin loop will actually interact either with the ribosomes or with intermediary proteins that interact with the ribosomes to help facilitate the RNA polymerase II to stop transcription and to actually disassociate from the DNA. It's a great question. Are there any other questions? All right. So we talked previously about splicing. We talked about exons and introns. Exons are the functional segments of the DNA that help encode parts of the gene for the subsequent protein. And the introns are these regions within exons that are going to be removed during the process of splicing. And so what that's going to look like is that at the end of splicing, what's going to end up happening is that each of the subsequent exons that make up that transcription unit, that make up that one gene, they're going to encode different domains of the translated protein. And so perhaps, for example, exon 1 could be the catalytic domain of a protease. Exon 2 might be the regulatory domain, and exon 3 could be the endonuclease domain or something to that effect. And so typically each exon usually represents a specific domain of the subsequent protein that it encodes for, and the introns are spliced out. And so splicing is what we're going to talk about for a couple of next slides. And it's the process of these introns being removed out to have to go from the pre-mRNA down to a mature mRNA with only the exons present in the mRNA transcript. And so the introns typically need to be removed. And when you look at, when you typically look up a specific gene and maybe look at what the primary transcript is going to look like, what's going to end up happening is that when you're counting the nucleotides, it's actually going to be quite interesting where perhaps the first exon will be 1 to 30, and the counting will jump over the intron and pick up on the next exon, so that when you look at the mature mRNA, it's only going to be a continuous count of the nucleotides present within the mature mRNA. So the splicing out of introns to form the mature mRNA occurs at these specific sites that we call splice sites. These splice sites are, as you can imagine, at the beginning and end of each intron, and have these two base conserved sequences that denote where these splice sites are. And as you can imagine, for example, the 5' end of an intron is typically a GU sequence, and the 3' end is an AG. As you can imagine, these are oftentimes, you're going to see GUs and AGs throughout an mRNA transcript. And so it's important to note that typically, based on what we understand about splicing so far, and we talked about this before, how alternative splicing can occur both in normal physiology, but can also happen times in diseases and different pathologies. And the way in which splicing events occur, we're still trying to understand. But what we do know is that for each intron, the 5' end, that 5' splice site, the 5' splice site is a GU and the 3' is an AG. Screen's gonna come back up in a second. It's on the back screen. There we go. and while the sequences after that can vary, the presence of GU and AG are 100% conserved across introns. And so this removal of the introns is catalyzed by a structure that we call the spliceosome. It's this multi-molecular complex of various proteins that we call small nuclear ribonucleic particles, or SNRPs. And so what this looks like is that typically if we have one specific intron that we're looking at that is sitting in between two exons, we're going to have our GU conserved sequence at the 5' end of the intron and the AG at the 3' end. And there's going to be what we call a branch point sequence that's going to be important during this process. So you're going to have the first SNRP U1. There's going to be U1 through 6. It's not important to remember which number does what part, but rather that SNRPs are a part of the splicing event and how it occurs. You'll have the first SNRP bind at the 5' end at GU, and the next SNRP bind at the branch point sequence. And it's going to recruit and promote the nucleation of many SNRPs coming together and promoting the looping of that intron. And as these SNRPs come together, what's going to end up happening is that the first cleavage point is going to happen at the 5' end. And it's going to loop that intronic region over towards the branch point and create what's called the lariat structure. All right. And after that, what's going to next happen after the lariat structure is beginning to be formed. And what's important to note is the figure is not entirely accurate. Upstream from the GU is not more intron, but this is all exon right here at the bottom left here. And so what happens is that after the lariat structure is formed and the 5' end is cleaved and brought over to the branch point sequence, what's going to happen next is that the 3' end is going to be spliced and the two exons are going to be ligated together, and the excised lariat is going to be degraded by exoribonucleases. And so this entire process is occurring across all the introns to form a mature mRNA transcript that's only constituted of the exons. Are there any questions? So this is the last modification event. We have the 5' cap, we have the poly A tail, and the splicing. Are there any questions about any of those processes? Yes.
I guess conceptually they kind of like, what are the differences between like getting out a very specific product splicing or a specific product transport.
So with eukaryotic DNA, with eukaryotes, you can have different forms. From one gene, you can have lots of different forms based on alternative splicing events. With prokaryotes, the way I understand your question is, are you asking, similar to polycistronic DNA, are there different splicing events that can occur for prokaryotes based on the multiple genes in tandem within the transcription unit?
Yeah, I guess like yeah, like just something like instead of like exons that are just parts of the genes.
Yeah, no, that's a good question. Because I've only worked personally with eukaryotes, I feel like my knowledge has always been focused there. With prokaryotes, this is me speculating now, I suspect with a smaller genome, the ability to create different splicing events based on these different genes in tandem is a way for them to have a higher degree of diversity within their genome, despite having a smaller genome than us. And so I suspect that the splicing of these polycistronic DNA permits wider diversity for them, despite having a smaller genome. Does that make sense?
Yes. So obviously, we also have
They do have introns. Yeah. Are there any other questions? All right. Well, thanks for giving me your time. That concludes today's lecture, and I'll be hanging back if you have any questions about the content or the assignment that's due tomorrow night.